Okay, I will share the screen. So I know how to. Okay, to share. Okay, does it work? Yes, sort of. So without the mask, I can wear the glasses and see something. So thanks to Katka for a nice introduction. I sort of modified slightly the title of the talk, which is Lousy and Wormy Tails, Drivers of Population Genetic Divergence in Host and Parasite Coevolution. So I will basically speak about lice and some other ectoparasites and worms and how they interact with their hosts on a population level and how this defines their genetic diversity. In my lab, we deal with several other topics. Aside the coevolutionary, we also work with invasive species, or generally the genetics or conservation genetics of some free-living organisms. We also work with marine parasites, which I will cover today, and also on arthropod-bacterial symbiosis, particularly with the group at the university with Václav Hybša and others. What we deal with most is basically looking into the mechanisms of speciation, or I would say genetic differentiation in general. And basically, if we simplify the situation very much, we have basically two models of differentiation. Either it's both are well known to everyone, of course, especially in this group, but we have allopatric speciation, where basically two populations are divided by some physical barrier limiting gene flow, or we have sympatric speciation, which expects some niche specialization within some group of organisms within a population, and this leading to the cease of gene flow and eventually speciation. In reality, these processes are much more complicated, of course. We can't always separate them into different categories, but what matters always is the gene flow between different units and the scale of effective population size. And these two things interacting together define how the speciation or differentiation process is running. And of course, which is again well known to this group, we have a ton of interactions in nature. So generally, the coevolutionary process happens at multiple layers. We have multiple interactions between hosts, between predators, between parasites, hyperparasites, and so forth. So anything I'll be talking about will still be a huge simplification, because I look mostly at the interaction between hosts and the parasites. In general, it's mostly parasites which are truly dependent on their host. Basically, the host is for them almost the whole environment where they can be found. And again, the parasites, broadly speaking about parasites, they constitute around 50% of the global species diversity, which means that any processes happening between these two counterparts are actually the drivers of the biodiversity globally. So it's an important question in ecology and evolution. And of course, then we're looking at processes like host specificity, specialization to different hosts, which might be limiting gene flow and starting speciation processes, but also historical co-diversification between the host and the parasite. And the populational level type of thinking in these analyses started when people realized that actually looking only at distant historical events is not always helping. So it was shown that host specificity, this specialization to one single host species, is not always a good predictor of co-speciation. So in the past, people expected parasites to follow the paths of their host. The host speciates into one or several species. The parasite will co-speciate as well. And it was shown that this is by far not always the case. So here comes an example from studying bird lice, where even in the same group of hosts, in this case it's doves, you can find ectoparasites which are co-speciating pretty well, even though there are still some host switches by these blue lines. But in general, the phylogenies do reflect themselves pretty well. But in the same host group, we find a different ectoparasite species where actually you see just almost only mismatches, only host jumps, host switches. So no co-speciation at all, even though the parasites still retain their host specificity to a high degree. So this led to the thinking that we need to look actually what is happening inside these species, particularly the multi-host species, how they differentiate between different hosts, what are the limitations of gene flow, and how all the processes started, not just looking at the finished species, so to speak. One of the great examples is always looking into human parasites, of course, because they gain attention, they're the most studied ones. And this was one of the inspiration works for me, for instance, was looking at the Helicobacter pylori bacteria, which is the stomach bug. Probably like 1/3, maybe 1/2 of us here carries it in their stomach, although asymptomatically. But it is in a small percentage of people, it is the causative agent of the stomach ulcers. And people looked at the genetic diversity of these bacteria, and they actually were able to see the same patterns of genetic diversity as in the human population. So these are different axes of principal component analysis of genetic linkage between human populations. And when they did the same for the bacteria, they actually saw the same pattern. So we see different historical colonization waves basically imprinted in human populations in Europe, but also in the Helicobacter bacteria. And not only that, when people look at genetic distance between populations, so it's like pairwise distances between populations across the globe. If you look at human populations, the further away you go on the x-axis, the higher is the FST distance on the y-axis, which is logical. The more distant populations you sample, the higher the genetic distance between them. But they found again the same in their stomach bug. They even found a very significant correlation in a local genetic diversity between the bugs and human populations. Usually, these populations here would be African populations, which are the oldest human populations, and they gained the highest volume of local genetic diversity. Somewhere here would be, let's say, South American Indian populations, South American natives, and the same again you see for the bug. So it seems like if you look at the populational level, whatever drives the genetic diversity of the host is imprinted pretty well in the parasite. And so this is humans. We're still one large metapopulation, basically. But I was always interested also in how this diversification is going on if more hosts are involved and host specificity can be plugged into the process. So my first example is from the Galapagos Islands and from Galapagos mockingbirds, which is one of the sort of inspirational species for Darwin and for speciation. It wasn't just the finches, it was also mockingbirds, which actually were the first ones where we realized that the Galapagos ones are different from the South American ones, and started his thinking like, all right, there might be something going on and species actually can evolve and differentiate. And with the Galapagos Islands, what we see is that the historical or the geology is basically producing the islands in sort of bands of different age because they're volcanic. So the youngest ones are here in the west, and towards the east we get older and older islands. So in theory, the biota should be stratified in the same way. With mockingbirds, we actually have 4 different morphologically or taxonomically recognized species in the islands. Only one species is pretty widespread, and it inhabits the youngest and the sort of mid-age islands. The older islands are inhabited by 3 other species. The oldest island is this one, Hispaniola. It was actually found out in the first molecular studies that the mitochondrial phylogeny doesn't reflect the traditional taxonomy. It seems like there's like a Western group of a Western lineage, then this, which reflects the species lineage pretty well, and then the Central Islands and Western Islands created 2 different lineages. So it seems like the phylogeny doesn't reflect the species recognition. And then we came up with the idea to have a look at the parasites, see how well their historical structure, phylogeography reflects that of their host. So we got data from samples from our collaborators and later also sampled ourselves. And first sequenced just mitochondrial genes at that time. So this is over 10 years ago when I was a postdoc. And compared the phylogeographies between the species. And sure enough, we found here in green color or grayish, very well phylogenies which matched very well, particularly for the old islands in the west. So these lineages were the same for 2 of the parasites and the host. But all the other lineages, even though they created sometimes island-only lineages in the parasite or the host, their relationships were almost unmixed, so there was no reflection. So there was a question, what could be causing these incongruencies? It could be either due to recent host migrations and then mixing the parasites, or it could be some historically old discordance. And so we looked at using nuclear DNA microsats at the current situation in the hosts. We found out that actually basically each island is its own lineage, and there's very little admixture, if any, is between the closest islands, like here, Santiago and Rábida. We see one individual which shows some recent admixture. These islands are just a few kilometers apart, whereas all the others are isolated by large bodies of water, and these birds do not like generally to fly across large distances. So we found out that actually each island is somewhat isolated evolutionary lineage nowadays, and that the discordance can be through recent admixture. Then we co-analyzed all the data from the host and the parasite in using star beast, as we treated it basically as if those were different gene phylogenies in a species tree reconciliation approach. And this allowed us to actually look at the historical discordance, which is simply because of general processes like lineage sorting, basically meaning that the nodes in the tree that we see in each of these organisms are older than the species themselves, which is a very common situation, of course. We already have some standing genetic variation in whichever population we looked at, and the relationships between alleles or haplotypes, whatever we define, are always older than the population if it starts to speciate. So all these tree nodes basically predate the true diversification of the species, and this is causing these incongruencies. So even though we see co-evolution and co-diversification between these parasites and the host, it's not always perfectly matched by just simple phylogenetic reconciliation. And when this star beast analysis was actually plotted against the islands, we saw a very good congruence between the mutual phylogeny, so to speak, between all the three taxa and the age of the islands. So depending on the age, we see different groups evolving there. And such approach also allows actually looking at coevolutionary rates, because it's well known for parasites that in general they evolve faster than their hosts molecularly. So in terms of substitution rates. And we actually saw that the lice had probably approximately twice the substitution rate of its host, which is common for most lice. In the mite species, we actually saw 9 times higher mutation rate. Again, the mutation rates or substitution rates in parasites are supposed to be higher for several reasons. One of them is they have often fluctuating population sizes, and the smaller the population size, the higher chance of fixation of mutation. And also their life cycle is several times faster than that of their host. So for one generation of a host, you get several, sometimes tens of generations of the parasite, and this also promotes the substitution rate. But we were also interested in general genetic diversity of the populations. So do we see co-diversification also in population sizes and in effective population size, the genetic determinant of the population size? And for microsatellites from the host, there were already published data by our collaborators from Switzerland, and they showed a clear correlation, and highly significant correlation between the size of the islands, which are the size of the habitat for the birds, and their genetic diversity. So this is the smallest island, which is just a few hectares in size. These islands are hundreds of square kilometers, and you can see clearly the rise of the genetic diversity of the population. And we actually found out the same for the parasites. So clearly, the larger the population size of the host, the larger the genetic effective population size of the parasite. So again, they do co-diversify very well. And nowadays we have whole genome methods. So Jakub Vlček, my PhD student, is resequencing the Galapagos mockingbirds, and we see across all chromosomes basically again the same thing. The smaller the island, like Champion Island, which is this little dot, which you might not even be able to see, has the lowest genetic diversity across the whole genome. So it's almost wiped out of any diversity, which of course may cause troubles in future adaptation. This species has actually died out on Floriana, and it only survives at least 2 small isolates here. Whereas on Isabella, which is the largest island, we see generally on these 3 big islands, we see the highest level of genetic diversity. So that of course the habitats can preserve, or more individuals can thrive here. It can preserve more diversity. But we're looking also at the diversity in neutral loci and in coding loci. So for instance, again, this is the clear correlation between neutral non-coding loci and the size of the islands. But surprisingly, in the, for instance, in major histocompatibility loci, in the immunogenetic loci, on a general level, in the populations, we also see this correlation between population size and the genetic diversity in terms of the total number of alleles which are preserved in the population. But we don't see this on an individual level. So here in the neutral loci, we see it even on the individual level. The larger the island, the more heterozygosity in an individual. But in these immunogenetic loci, we don't see that, which is telling us that there are other processes which actually can preserve diversity on an individual level, because this pressure from the pathogens is probably removing hosts which are doing very poor, which have only a few copies of different immunogenetic alleles, and there is selection which is preserving some diversity there. We're also following this MHC story by actually trying to link the genetic diversity with the parasitization itself. So we have these 3 species of ectoparasites, and we're looking at different variants of MHC alleles, how they are shared between islands. So these are the 4 species. Some alleles, most alleles are very well shared within populations. Some are shared also across populations. But we were able to show that actually there are 2 MHC supertypes. So this is like functional types of the MHC molecules, which are very well correlated with abundance of one of the parasites of Mercidia laus. And this was the first time anyone found in free living bird populations any type of association between actually an ectoparasite and an immunogenetic locus. There are more parasites which we didn't look at yet. For instance, these birds contain also intestinal parasites, coccidian parasites, which in collaboration again with people from the faculty, we're trying to look at now what is the species diversity, and maybe this could be linked again with some of these functional genetic differences. Okay, so this is about a pretty simple scenario where we have islands which somehow differ in their history, in their size, and all the fauna is basically affected by that. But what if we have more hosts and more complex geography involved in the co-evolutionary process? So we also work on these organisms, which are mice, wood mice from Palearctic. Mostly we focus on Europe, and on their ectoparasites. And we started with looking at this louse, which is called Polyplax serrata. And in the first study, just a very brief introduction into the system. So actually, Central Europe is a very good place to study these organisms, because all or several different species of Apodemus mice overlap in their distributional range here. So we can actually sample sympatrically several species and then look at their parasites. We also have a very good picture about their historical phylogeography. So the European mountains were always the limit in distribution ranges when climate shifts and the species retreat into refugia. Most of the studies actually disregard completely this mountain range, which is the Czech one. And I will show you that they shouldn't do so. So even these smaller mountains played some role historically in this process. From literature, we know very well that there were several refugia for these species of mice in different parts of either Southern Europe, like in this case of this Apodemus sylvaticus, or in Eastern Europe or Southeastern, in the case of Apodemus flavicolis, which has 2 clades with different distribution. But both of these species, after the glaciation receded, they recolonized Europe from the south. Some of them maintained even some genetic gene flow continuation, even when they were separated in refugia, because all these shallow seas weren't present at the peaks of glaciation, so there was some gene flow maintained. So what we did, we sampled across Europe all of these different Apodemus species. Then we looked again at the genetic relationships of their lives. And we actually found out that there is a strong link between different genetic clusters and different host associations. So we found one lineage which we called non-specific, that's why there's N, because it infected 2 hosts, Apodemus flavicolis, which is the yellow-necked, and the sylvaticus, which is wood mice, I think, across the whole Europe. Then we found another lineage which was solely specific to one of these 2 species. So actually, these 2 lineages live in sympatry, but one has wider host specificity and the other one has a very narrow host specificity. And then we found a 3rd lineage, which is only found in Eastern Europe, basically, and it extends to Central Europe, but it's not as common. So we further focused on these 2 lineages because they live in sympatry and differ by the level of their host specificity. So we were interested in how this is reflected actually in their population genetic structure and diversity. And first, using microstats, we verified that actually what we see in mitochondrial phylogeography is reflected in their nuclear genetic relationships as well. So we recovered the same groups. And then we wanted to test one of the known hypotheses in coevolution and in parasitology, which predicts that basically a monoxenius parasite, which is a host-specific parasite living only on one host, should show deeper population structure than multi-host parasites, for the simple reason that the more hosts you have, the higher the opportunities for gene flow and migration, the larger effective population size as a result. So you can retain more genetic diversity and be less stratified geographically. So we compared diversity in these two sympatric lineages, one which is the highly host-specific, only single host, the other one which still for a parasite is pretty host-specific, but having two hosts, so basically far more opportunities to disperse. And we actually found out that across the two lineages, we saw smaller genetic diversity in the specific parasite and in the non-specific, and also higher FST, the population structure, much more significant pronounced than in the non-specific parasite. So again, this reflected very well in this hypothesis. We also tested it locally, so looking at pairs of populations which were sampled sympatrically or almost sympatrically in the case of Italy here. And again, consistently, we always found a lower value of genetic diversity in the specific population than in the non-specific one. And also, when we just looked at the isolation by distance pattern across Europe, which reflects population structure very well, we always saw a higher volume of this in this non-specific lineage single host than in the two host one. So is this a proof of Nader's hypothesis? Sure, might be, potentially, but the problem is that there are always more processes involved, and also historical processes has to be taken in account. So actually, by more deeper sampling, we found out that this host-specific single-host lineage contains two different mitochondrial lineages with different distribution in Europe, which might be further limiting their nuclear genetic diversity, which we looked at. And again, we saw actually it correlates very well with some of the mountain ranges in Europe. And as I said, there seemed to be some historical effect of these Czech border mountains, because basically the two lineages were separated by the Alps and then by this and maybe the Carpathian range as well. We found out that there actually is a contact zone in western part of the Czech Republic. We didn't see any such contact zone in the non-specific lineage, where it was much more chaotic, the distribution of clades across Europe. We found out that this pattern is not by the host. It's not like that both the parasite and the host would create lineages which would meet somewhere here and then hybridize. So the parasite, the host actually showed to be panmictic across this area. Then we thought that actually there might be something going on with the mitochondria themselves, which is why we see this pattern, because we didn't see this pattern in microsatellite DNA at all. It seemed like we can identify different populations, but we don't see this strong breakage anywhere here. So we thought maybe it's some role of mitonuclear incompatibility going on, which is quite common in nature. Particularly lice are weird because their mitochondrial DNA is not in one single molecule like in most animals. It's actually broken up into single genes which create their own mini chromosomes or mini circles. And then theoretically, if there was a mutation in one of these, it wouldn't affect all the other, but it could affect the incompatibility with the genome, with just one gene. So we wanted to look at that, and So we also, it could be of course by endosymbionts, because these lice have endosymbionts which help them with digesting blood, and because blood is depleted in some vitamins, for instance in vitamin B, so they have endosymbionts which again are helping them, and they have to somehow genetically communicate with the host. So we did whole genome resequencing of 28 lice across this zone of contact, and we actually found out that both the endosymbiont and these mitochondrial mini-chromosomes show the same pattern, like two different lineages. But in the SNPs from the nuclear genome, we found out hybrids. So we saw that actually this is a true contact hybrid zone, where again the host is able to freely pass this zone, so it carries the parasites to short distances. Of course, this host migrates only a few kilometers within its lifetime, but it doesn't carry the parasites across this zone of contact, and there's a typical hybridization happening with the hybrid zone in the center. And we're still following this question, like whether this is just internal genomic incompatibilities between the two hybridizing lineages which are keeping this, or whether there are other mechanisms which could be involved. But on top of this, we're looking at other parasites from the same host, from different mice across Europe, and repeatedly, this was work by my master students, repeatedly we're finding similar patterns, where actually, whether we look at east or west, we're finding different lineages with, I won't go into details here, it's not needed, but with different frequency of distribution. The most lately, we looked at one of the mites, which is living actually on all of these different hosts. So it's not host specific at all. We found out that there are different genetic, again, lineages with different distribution or frequency of distribution in Europe. Again, between east and west, there seems to be some sort of differentiation happening. In the east, the populations are much more panmictic in this lineage, in the green lineage, whereas in the west and southwest, there's this red lineage, which is much more structured locally. And we hypothesize that this is due to different historical phylogeographical history of the regions, with smaller, more fragmented populations historically in the west than in the east. So the coevolution runs long, runs deep, and it can create different patterns in different organisms, even parasites sharing the same host, like in these three cases. So I will break into the last bit of my talk. I'm sorry if I'm too quick. If you still feel like you want to ask anything right now, just do it. Otherwise, hopefully we'll have time for some questions at the end. And the last bit is about endoparasites. So actually, ectoparasites were historically always, or by researchers, thought as one of the best models for coevolution and co-speciation, because they complete their whole life cycle often on just one host. Whereas endoparasites, like tapeworms, for instance, they switch several hosts in their life cycle. They have to adapt to each of these hosts. And of course, there's much more potential for dispersal, for connecting the populations and so forth. And in my research, we focus on this tapeworm called Ligula intestinalis, which has a 3-host life cycle. But we're most interested in the fishes, because this is where the parasite spends most of its time, about 1 year, to develop into these really, really large tapeworms, living freely in the body cavity, not even in gut. So it's a larva which castrates the host, and it basically draws all the energy into building up a huge biomass. Then the fish is eaten by this bird, and inside the bird, actually, there's nothing else happening than just maturation of the tapeworm, releasing the eggs, and within 5 days, it's gone. It doesn't harm the bird at all. It doesn't basically take any energy from it. And then in water, there's a larva which hatches, infects some copepod, spends about 2 weeks in this copepod, and it's eaten by the fish, and that's how it runs. So we're focusing mostly on the fishes because they seem to be the most important part of the life cycle, where all the energy is being taken. And this parasite has a global distribution, some mostly infecting cyprinid fishes, but also some other, even some really nice ecological examples of parallel evolution. So in this case, this galaxiform fish is not related at all to salmonids, but if you looked at it, you would thought this is a trout, right? So this is from the southern hemisphere. It's an example of an ecological alternative to northern hemisphere trouts, but it's evolutionary unrelated fish. But these also can be infected by this tapeworm. So we collected tapeworms from all around the world. We actually saw that almost each biogeographical area creates its own genetic lineage, but in some areas we saw several lineages living in sympatry, like here in Europe. We have a cyprinid lineage, or which we call derived cyprinids, and then a lineage which infects several other cyprinid taxa. They're genetically differentiated, but they share the same definite hosts where they breed. So they share actually the same fish-eating hosts, but they do not interbreed. So it's two lineages historically already diversified. Looking at, again, at the time, microsatellites, we found out the same picture with two lineages in Europe, and then in the rest of the world, the same cladogenesis, basically. But we were most interested in the European situation. We actually found out that this European lineage from cyprinids is found also in Northern Africa, where these cyprinids were only introduced, so they're not native. And we found out that we already see a genetic differentiation between Europe and Northern Africa only after 40 years when this fish was introduced there. We verified this with a coalescent analysis with some modeling, which showed us that the recent gene flow was only one way. It was from Europe to Northern Africa, and it must be associated with French people building actually drinking water reservoirs in Tunisia, and then they had this commercial sort of plan to plant the cyprinids there. The cyprinids would be food for predatory fishes, and the predatory fishes would be exported back to European fish food markets. The whole economical plan collapsed, but still the European fishes are there, and they're being still infected by actually European originated parasite. So this is just a detour, but the most important question that we were asking is that in Europe, we didn't see, using the microsatellite DNA, any genetic differentiation between the populations at all across all Europe. It seemed like one convicted population maintained by this short-term bird migrations, but still, even if the worm is kept in the guts of the bird for 5 days, the bird can fly far away and probably maintain one large population. But this was in sort of disagreement with what we know ecologically, because we know that actually in Western Europe, it's mostly breams being, in Eastern Europe, it's mostly breams being infected with Ligula. In Western Europe, mostly roaches, and in some parts like Central Europe, they seem to kind of overlap. So two of my students looked at this situation, master student Pavlina and now Masoud, who's continuing this research. And we genotyped using RADseq, we genotyped hundreds of worms from all across Europe. And sure enough, we actually found out two different population lineages, which are very, very closely related. So we see only a faint population structure between them. And across Europe, bream infecting fishes contain one sort of lineage or population. The roach and bleak infecting worms are another one. We only found a single case of host switching, where the worm belonging to this group basically was infecting this host. And we went even further. So we questioned whether this is a case of adaptive diversification. Is it due to the host? And actually one generalist parasite now in this real time basically diversifying and adapting to two different hosts. So we actually looked at a population sampled in these two lakes in northeastern part of the Czech Republic, where all hosts were present in both lakes. And even at this small scale, we saw the same thing. Basically, this is a PCA plot of single nucleotide polymorphism diversities. We saw that the bleak and roach population created one cluster. The bream population in both lakes created another cluster. We had some outliers, which we still have to look into, but most of the diversity explained was on this principal component 1 axis, so it runs in this direction. And then we also looked at loci, which could be actually causing this differentiation by the so-called FST outlier approach. So we just compared these two groups of individuals between themselves and looked at loci which are more significantly further apart than they should be if it was just a random genetic differentiation. And we identified about a few dozen of loci, but now we're in a situation where we have just assembled the genome of this parasite because it wasn't available before. And we have a true high resolution, nicely resolved genome. We have the first transcriptomes, so we're looking at differential expression of genes which might be causing this. And we see actually, looking at the two hosts, we see some transcriptional differences. And we also started with fish immunogenetics to just look at if we can find any interesting genetic variants in these two fishes which differ in relation to parasitization by the worms. So this is the situation where we got to. So we think it is a very promising model for sympatric ecological speciation, like in the case of, for instance, sticklebacks, which if you look at lake populations or stream populations, you can find different morphotypes, and these morphotypes were very well linked with different genes and the diversity of these genes across whole chromosomes. So our aim is then to map the diversity from SNPs on chromosomes in Ligula, trying to search for these islands of genomic diversification and to look at if interesting genes lie nearby, if they could be the adaptive genes that allow sympatric diversification of the parasite. So I went quick, so just to wrap this up, hopefully you're not too tired. So basically, as I started with these two potential mechanisms of differentiation and speciation in nature, where does host specificity stand in here? And so is host specificity leading to these adaptive sympatric diversifications? Well, we think it might. At least in this last example of this Ligula tapeworm, but we're collecting more data to find out the true genomic mechanisms which might be then involved to basically test this hypothesis. And is allopatric diversification related to host specificity? And yeah, we think definitely it is in many cases. So if you get geographically isolated hosts, and if different host species happen to be under different geographic circumstances, the parasite has a chance to speciate, and if later they become admixed, the parasites do not admix and they retain their genetic identity. And we see this across basically all of these examples, even in Ligula, where if you look at the global situation, you see each geographical unit contains its own worm, but only sometimes they also live in sympatric. So again, it must be the mix of the two, but it depends on the spatial and historical level that we're looking at. That's it from me. I think I had 45 minutes, which is just about. There's a number of people who were involved in this from different institutions which we collaborate with, but mostly most of this work has been really done here in collaboration with the students in my lab and with people like Václav Ivša from the university. And with that, I'd like to thank you all for your attention, and hopefully we have time and energy for questions. So anyone would like to ask anything, and any, whatever, either something weird you spotted that doesn't really play or anything. Yeah, go ahead.
Yeah, thank you very much for a nice presentation. I'm just wondering, it's maybe a little bit out of the main topic, but you mentioned the Ligula species being like cosmopolitically distributed. Is it just one species or like different species?
Yeah, that's a good question, because there are like 3 traditionally described species, but with very uncertain morphological traits to look at. And one species, which is this Ligula intestinalis, was supposed to be truly globally dispersed, whereas 2 other species were mostly found only, one of them only in the Eastern Palearctic, the other one only in Western Palearctic, basically. What we see is that these 2 species with local distribution, they are probably true species. And again, and in Ligula intestinalis, that's why we call it sensulato also. It's probably several species with different width of geographic distribution, but some of them are really very wide. So like this European clade, which I think is a species, is in the nearly whole Palearctic, and also in some parts of Southern Asia. And then most other, like in Africa, we see 2 other lineages and elsewhere. So it would probably be a assemblage of different species. Yeah.
It's like most jelly gulls and the grebes, or even some, let's say, birds of prey, because just osprey as a bird of prey is probably like cosmopolitically distributed.
Yeah.
It like transfer it all over the world. Otherwise, it would have to switch the hosts.
It would, yeah. So it can actually mature in any, I would say, in any vertebrate. So experimentally, if you infect even a hamster, it will mature inside, because it's not using the resources. It's just using the heat basically from the host to mature. So any fish-eating vertebrate could be a host. The thing which is limiting for the distribution is the time, because it only takes like 3 days to mature, then 2 days for breeding or releasing the eggs, and then it just disappears. It vanishes from the gut. So within those 5 days, wherever the host can get to, the parasite can, in 1 generation, basically. So that's why it's not entirely global. Like each of these lineages then could be, if it was with ospreys, yeah, it could travel anywhere, but it does not. And we actually found out that it has even problems crossing the Mediterranean Sea, for instance, because this European lineage, and as I spoke about it, like recently it was introduced to Northern Africa. If the birds would easily carry it across the Mediterranean or frequently enough, it would have established itself earlier in that. But and we would have also seen 2-way migration patterns in the gene flow, but we only saw a single-way gene flow, the one from Europe to Northern Africa. So we think that yeah, this is the limit. Like it only takes a couple of days for the bird phase to live, and then it disappears. And within those 5 days, it has to find a freshwater body which is capable of maintaining the next population. So there has to be the crustacean host, and there has to be also the suiting fish host. So that's, yeah.
All right, thank you.
Anyone else? I don't know if Katka can still hear us.
Yes, I can still hear you. I'm just checking. There is no one online asking anything.
Okay.
But I would like to have a question which is more like ecological, because there is big literature about antiparasitic behavior of birds.
Yeah.
But I don't see too much of the literature on the mammals. What do the mammals do actively to avoid the parasites?
Yeah, it depends on which parasites. I wanted to put you on the screen, but I didn't. But
Okay,
you still have
yeah.
some.
I can put myself on the screen.
Okay. So if we speak about ectoparasites, and that depends whether you mean to avoid or remove. So I assume you mean truly to avoid being infected rather than
yes, maybe avoid to be infected.
Yeah. Well, I must say I can't really say I studied this deeply in reading literature, but basically any type of for the foodborne parasites, so any type of being aware of what the organism in question eats is one of the ways, and you can see this even in human populations. In many human populations, you have taboo on some sorts of food, and it is right. It is hypothesized that historically this was due to bad experience with either some pathogens like bacteria or parasites, and so that's one case. With the other, I'm not sure whether there are some sort of long-term evolutionary strategies like living in smaller groups may prevent spreading some parasites or diseases. But yeah, I'm not really aware of anything. But you can, yeah, there's someone asking.
If I may join, I don't recall anything like that as well. Maybe in terms of primates, we know that monkeys like chimps, apes, they build every night
A different nest. Yeah.
Or maybe bathing in the sea, in salt water, maybe away.
Yeah, there are ways of removal, of course. So in case of mammals and ectoparasites, and it's the same for birds, of course. Removing ectoparasites is one way. It's also a way of social bond, even in some bird species. So that's one. But when we're speaking of avoiding the infection, I'm not sure whether there would probably be people having a clear answer. I don't.
Okay, I will try to check somewhere. Okay, thank you. And my another question, if there is no one else, was about those, whether you know something about the nightingales, the two species of nightingales which have the hybrid zone in Poland. Do you have any idea how is it with their parasites on the contact zone and the hybrids?
I know that I think it's people in Prague who are looking at this, but from the point of view of bloodborne, I'm not sure whether it's Plasmodium or hepatocytosome, whatever it's called. So I think they're looking at this, but no, I don't know if there's anything clear coming out of that. So
okay.
With birds, of course, it's different, especially with seasonal migratory birds, although they kind of tend to come back to their homeland. But because they, especially with some parasites, they actually often do not necessarily acquire them in the place where you study them. Like they might acquire them, during their migration routes to Africa or wherever. So it's more complicated, but I don't know actually how these studies, whether they turned out anything. Yeah.
Okay, yeah, that I didn't realize. Yeah, that's of course they might have completely the same parasites because they get them on the migration way somehow.
Yeah.
Okay. Okay, thanks.
We're actually looking at, we have a project in Vietnam where we're looking at 3 sympatric species of passerines and looking at their Plasmodium and hepatocytosome, but this is still basically we just got to the sequencing phase now. So we should learn soon if we see any differences in local fauna of the bloodborne parasites, and we're also trying to link this with the immunogenetics of the host. So yeah, but yeah, we don't know yet.
Okay, thank you. I don't know. I don't see anyone asking from the online people if there are some questions in room.
Seems like, yeah, there's one question.
I was wondering if you also try to see the tapeworm genetic diversity linked to the number of hosts. Is it possible to try to
the number of hosts, like the population size of the hosts, you mean? We didn't try to link this. It would be, well, in some cases, you could imagine that the thing is that where we see the differences in the local, potentially adaptive or whatever diversity, this is the two most common fishes in the Czech Republic, probably. The breams and the roaches are ecologically very adaptable, and you can find them in almost any lake in large quantities. So actually diversify between, if we can have a good estimate of if there's any difference at all in the population size, it will be still very large population sizes. And but we're definitely looking at the genetic diversity of the lineages of the parasite. Again, they generally have a huge effective population size, any of these tapeworms. So they might differ slightly, yeah, but that's hard to link with anything like this. But I agree, it's a good idea, because if you do see some stark differences between the populations of the host, it might be reflected there as well. Okay, it seems like it, probably. Thanks again for your attention.